Dr. Farah is a Fellow of the American Academy of Arts and Sciences, a Guggenheim Fellow, and the recipient of honors, including the National Academy of Sciences, Troland Research Award, and the Association for Psychological Sciences Lifetime Achievement Award. She has spent her career exploring many topics within cognitive neuroscience. Her current work focuses on neuroethics, the ethical, legal, and social implications of neuroscience. Martha, we're delighted that you could join us today. Welcome, and why don't you start, and then I will introduce the next speaker with his correct full name.
All right, well, thank you very much. I'm very honored to be here and very excited to know that the Commission is starting to think about neuroimaging. It's an important topic, and I hope I can help. Let's see, where do I point this? Okay, so in overview, what I want to do is discuss some of the ways in which neuroimaging raises new issues and poses new problems relative to earlier technologies, including genetics. Then quickly go through some of the main ethical and societal concerns about imaging and sort of triage them for you, put them in different bins that I think require slightly...
Can people hear in the back? You can hear? Okay, good.
Okay, great, thanks. After a talk I gave once, a group came up to me very excitedly. I thought, oh, they're going to tell me how much they like my talk. And they said, we're speech pathologists. You need professional help. So I managed to be inaudible even when I'm mic'd. I'll try to speak clearly. Okay. Okay. Go through some of the sort of well-known and less well-known concerns about imaging. Give you a taste of what's out there and what's coming by presenting two examples of, I think, currently problematic applications of brain imaging, and finally some concluding thoughts. Okay, so what's new? What's societally and ethically new about imaging relative to other technologies, most notably genetics? Well, in behavioral genetics, what we do is does this work? Yes. We relate people's genes to their behavior. And the field has been successful and found some correlations. You know, we have biomarkers for various kinds of disease states versus health, for personality traits, intelligence, as was already mentioned. However, this approach leaves out the effect of lived experience on behavior, which is substantial. By focusing on the brain here in the middle, we are looking at essentially what integrates the effects of genes and experience, which in fact, you know, interact in much more complex ways than are shown by just, you know, two separate arrows there. But through their interactions, they build a brain, and the brain drives behavior. So by looking at the brain, you can see in its position here on the graph that we are looking at an organ that encompasses all of the antecedent causes of behavior, and interestingly, which is one causal step closer to behavior. And that suggests that we might actually get some additional predictive power out of brain imaging versus genetic analyses. And that turns out to be the case. So we already heard mention of the fact from a previous speaker that personality traits do to a certain extent correlate with specific genes. The correlations are small. Usually less than 5% of the variance in a trait is accounted for by a single gene, usually more like 2 or 3%. In the most kind of garden variety fMRI studies, it's more like 25 to 35% of the variance in personality traits can be accounted for by patterns of brain activation. So that's an order of magnitude. In addition, another thing that we should take out of this diagram is that unlike genetic information or overall life experience, by looking at the brain, we can read, we can understand states, mental states as well as mental traits, not just long-term personality, intelligence, whatever, but also current mood, intention, am I lying, do I want to buy this thing? That all comes from brain imaging. It's in principle available, and in practice available as well. So I want to just talk about some of the upshots of this diagram. What are the distinctive characteristics in terms of societal implications? Relative to genetic testing and genetic research, brain imaging gives us a more sensitive measure of the causes of behavior, that order of magnitude. It reflects environmental determinants of behavior, which are important determinants. As a result, it captures learned psychological traits. A trait like attitudes towards other races. There's been quite a bit of research done on this in cognitive and affective research. It is a learned trait. It's unconscious, or a component of it is unconscious, and it correlates with patterns of brain activation. It captures psychological states as well as traits. Again, this is what lying or neuromarketing, lie detection neuromarketing is based on. And, didn't mention this before, but it suggests a broader range of targets for intervention, to not just, say, okay, now we see how this person's brain is working, but now we can figure out how to change the way it's working. Pharmacologically, with transcranial brain stimulation, with deep brain stimulation, et cetera. Relative to psychological testing and research, because that is a technology, if you will, that addresses some similar questions, it is often more sensitive. I won't talk about that now, but we can maybe talk later about some sort of head-to-head comparisons there. And finally, the information sought in a brain imaging protocol for something like personality or racial attitudes may not be evident to the subject who's being scanned. So in both of those cases, the protocols that have been used just involve looking at pictures of people's faces. So there's nothing in the task itself that says they're looking to measure your personality or your racial attitudes. Okay, so what are some of the problematic characteristics? Again, problematic in their societal applications. I'm going to sort of do a triage here. Familiar concerns, more novel concerns, and within the more novel concerns that we've heard a lot about already, maybe a little exaggerated, and some concerns that I think we really need to pay attention to. So first of all, the familiar concerns, which I want to emphasize are not unimportant concerns. It's just that the good news is we have grappled with them in other fields, including with genetics, and as the previous speaker was saying, with other kinds of biomarkers. The first one, there we go. The first one is, the validity of any given measure across ages, levels of socioeconomic status, cultures, et cetera. Almost all the brain imaging research that's done is done with college undergraduates. There is reason to believe that in some cases, the results may not generalize. The precision of prediction may not generalize. Incidental findings, another very important issue. It certainly acquires new wrinkles in the context of brain imaging, but basically there is plenty of bioethical precedent available to help us grapple with this, and we'll hear more about this later, I know. And similarly, privacy of records. Brain imaging, like genetics, it gives you information. When you're acquiring the image, you may be acquiring it for one reason, but it contains information that can then be later used for other reasons. And so, but again, we've been around this block. Isn't to say we've solved all the problems, but they're not totally new. There are some problems that are specific to brain imaging that have been discussed a fair amount in the neuroethics literature that have a kernel of truth to them, and I don't want to cross them off our list of things to think about, but I think they have in some cases been a bit overdrawn. One is the idea that this is going to lead to mind reading, and this gets back to the question that was asked earlier. Okay, you can see that somebody's looking at the letter M. That's not really mind reading. Can we put Hank in a scanner and find out that he's thinking, every time I've spoken with Martha, she's run overtime. I wonder if she's going to go overtime again, and then I'll have to cheer her up afterwards because she'll be feeling bad. And we are not in our lifetimes. Don't need this again. In our lifetimes, we're not going to be able to do that. I rarely make definitive statements like that, but I'm willing to make that one. Nevertheless, there is some non-trivial, personal, cognitive, affective information that can be read from brain imaging beyond just what letter you're looking at. And this is largely driven by some of the very exciting new pattern classification statistical techniques that have been used. We can talk about those more later too. A sort of familiar refrain is, oh, they're not even imaging the brain, they're looking at blood flow or oxygenation. I mean, yes, but the point is it's a measure that is not perfectly, but quite well correlated with brain activity. Astronomers, when they want to know the chemical composition of a star, don't fly out there and bring some stuff back in a test tube. They look at absorption spectra. The tool doesn't have to be transparently related to the subject of study. Statistical voodoo. There is a lot of statistics that intervenes between the acquisition of the data on a scanner and the scientific inferences. And I think Bruce didn't have time to go through this, but it's really a lot of processing. There's so much processing that they divide the processing into pre-processing and processing, but the pre-processing is processing, okay? There's a ton of statistics. They're done for good reasons. They're done to more accurately and validly make inferences. But all that statistics arouses suspicion in some cases, and people sometimes make mistakes. And the paper that was so helpfully named Voodoo Statistics a couple of years ago, criticizing a certain group of papers in the social neuroscience field, sort of underlined this fact. But the point is, we know how to do statistics, we're getting better, and it's not an inherent problem with the method. Finally, there is the worry that images are inordinately persuasive, that you tack a brain image on any crazy claim and everybody believes it. And I think that is overdrawn as well. Okay, so some more new challenges. First, there is, kind of related to the last, there is limited public and policymaker understanding of brain imaging. They look like pictures, but they're inferential, they're not photographic. And what that means is that if you don't know, what task was being used, what contrasts were being analyzed, and so forth, you don't really know what the image means. So it sort of invites misunderstanding. There's also a very common reaction that if it's in the brain, sort of biological, it must be innate. And if it's innate, it must be immutable. And of course, taxpayers are not so happy about investing, their money, resources, in trying to change or improve things that science has shown are immutable. Okay, and we have the whole bell curve mess to refer to there. So brain does not mean genetic. Remember that first picture? Enough said. And finally, brain images are no more real than behavioral evidence. For some purposes, you may prefer behavioral evidence for legal purposes. There was a big brouhaha recently about a PET study that showed that cell phone use activates the part of the brain near the ear. Well, yeah, I suppose that might make you think again and look more carefully, but the relevant data is the epidemiological and the animal experiment data. If people aren't in fact having brain diseases, then who cares if the PET scan activates, Okay, reverse inference.
Martha, you have 1.5 minutes.
Okay,
So just
I am gonna skip
Thanks.
I'm gonna skip there, and I am going to just say lie detection here now. It is it's, in my opinion, its validity has not been demonstrated. It has many potential uses, including national security, law, personnel screening, and so forth. There have been 2 attempts to use it in court. I was at the Daubert hearing in Memphis. It was much closer than I ever would have dreamed. Diagnostic imaging and psychiatry. Clearly, imaging plays a vital role in psychiatry research, but there is a broad consensus that it has any role to play at this point in diagnosis. Yet we have Daniel Amen's clinics and several others saying, you know, my testimonial, they imaged my brain, and now I am better. So to summarize, what we have here is a combination of wishful thinking, and I want to emphasize that there really is great good that can come from this, okay? There are a lot of problems in society that come down to needing to better understand human behavior. And brain imaging is helping us to do that. And in terms of the question I got from some of the staffers, you know, what's the time frame? I mean, right now, brain imaging is delivering useful information to neuromarketers. I would be surprised if in 10 or 20 years it was not delivering useful information for psychiatric diagnosis. And I would be surprised if in 10 or 20 years, assuming there is research being carried out during that time, we don't have a pretty clear answer of whether or not it is useful for lie detection. But right now, we don't have the science to back these things up. All we have is the allure of science. Oh, it's scientific, it's objective, and so forth. And you combine that with the profit motive, and we start pushing these technologies out into the marketplace before they're ready. We don't know enough about the validity and accuracy, and so there's great potential for harm. Can I give you one more slide? Okay, so concluding thoughts. I think it's important to view the challenges and the promise of fMRI in the context of something broader, and that is the final coming of age of cognitive and affective neuroscience. Not just that we can image the brain, but we can understand it and we can control it and improve it. In addition, this growing tendency of lay people from all over to think of themselves increasingly as their brains. So I think it's worth bearing that in mind.
But Martha, if it's one slide,
okay.
Gotta go quickly, because thank you.
Promising new technology, again, I've sort of focused on problems. Bioethics tends to bring that out in people, but there's a lot of good a lot of problems that can be solved. It's the premature uses that are bogus and potentially harmful. And for policy, I want to point out that policy does not equal regulation. I think the thing to do here is shine a nice, strong light on these applications. It can be very sanitizing. There's little here that couldn't be corrected by sort of saying, look, here's the validation studies of lie detection. This would also hasten the day that we have more socially beneficial applications of neuroimaging. And I want to say that if we don't pursue this stuff, if we, in an effort to protect our citizens, try to regulate it away, other nations will develop these tools. And I think, the US should be helping lead in the science and also in the ethics. Thank you.
Thank you very much. Thank you. Hank Greely is the Dean F. and Kate Edelman Johnson Professor of Law at Stanford Law School and a Professor of Genetics at Stanford University School of Medicine. Previously co-director of the Law and Neuroscience Project centered at the University of California, Dr. Greely is the current chair of California's Human Stem Cell Research Advisory Committee. He also chairs the steering committee for the Stanford Center for Biomedical Ethics and directs Stanford Center for Law and the Biosciences and the Stanford Interdisciplinary Group on Neuroscience and Society. Many hats, Dr. Greely's research focuses on the implications of new biomedical technologies, especially those related to neuroscience, genetics, and stem cell research. Welcome, Hank. We're looking forward to hearing your views, and it's clear that you bridge the broad areas that we're discussing today. So thank you.
Thank you, and I also don't use slides, so I'm just a talking head here. Thanks for the opportunity to do this. It seems to me that trying to figure out how best to aim the public resource that is this commission its staff, and the public attention and respect that its findings will get is really intellectually a very fascinating and difficult problem for me. For you, I'm sure it's a fascinating, difficult, and very practical problem. So I hope to try to help you with that a little bit. 3 caveats. First, there are 3.4 billion base pairs in a haploid human genome. There are probably 3.4 billion ethical, legal, and social issues. I've got now 14 minutes and 17 seconds. I'm only going to talk about 3 of them. That doesn't mean that there aren't a lot of other good issues. In particular, I am sorry that I'm not going to focus on the forensic and other non-medical uses, which I think might be a very good field for you to explore. Second caveat, much of what I'm going to say will be things that have been able to be done before, but they haven't been done before to the same extent. I think this is one of those areas where differences in degree become differences in kind. It's like automobiles. In 1900, there were 20,000 autos in the world, and they were curiosities. By 2000, there were 500 million autos in the world, and they'd changed the planet. So I think a lot of what we're doing here, a lot of what we're talking about, similarly, is change in degree leads to change in kind. Let me go straight now to the 3 things I want to talk about. Non-invasive prenatal genetic diagnosis, clinical use of whole genome sequencing, those are 2 types of predictive test, and then bridging and combining those along with some of the other things we'll hear, both on genetics and neuroscience, issues of how to deal with the data derived from those kinds and other kinds of tests. Non-invasive prenatal genetic diagnosis. We've been able to do prenatal genetic diagnosis for 40 years now. It's a technology that's been around for a long time. This past year in the United States, somewhere between 1% and 2% of pregnancies involved prenatal genetic testing through amniocentesis or chorionic villus sampling. So not very many, 1% to 2%. The rate limiting factor has been the difficulty of getting DNA samples from fetuses. Fetuses are carefully protected from all sorts of outside influences, and it's not easy to get their DNA. Amnio and CVS are not fun, pleasant, cheap, or entirely safe procedures. My wife had one of each. She didn't like either of them. It turns out, though, that there is a lot of fetal DNA to be had in the bloodstream of the pregnant woman. The DNA is chopped into very small pieces, but by the 5th week of pregnancy, 5 to 10% of this cell-free DNA chopped up in the woman's blood serum is from the fetus. The rest of it is, of course, her own DNA. It is now possible to do clinical testing of the fetus with that DNA based on not an invasive procedure, but a simple blood draw, 10 milliliters of blood. It looks like it can be done as early as the 5th week of pregnancy. There is no risk of miscarriage. The procedure itself is simple and cheap. In fact, it probably doesn't even require another phlebotomy because pregnant women getting prenatal care have their blood drawn a lot. So it's just one more tube of blood drawn. This is currently in clinical use in Europe for Rh factor determination. If the woman is Rh negative, you're interested in whether the fetus is Rh positive. It's in very limited controlled clinical use in Great Britain for sex determination, when the sex would make a medical difference, typically when there's an X-linked disease, so you're worried about a male fetus. 2 recent large trials were published within the last 2 months of its use to detect aneuploidies, particularly trisomy 21, which leads to Down syndrome. Both showed accuracies in the 98, 99, 100% range for sensitivity and specificity. All that is interesting. All of it is being driven by increased whole genome sequencing or increased sequencing ability to shotgun sequence all these little bits of DNA and then put them back together. The real payoff is, I think, 2 to 5 years away when people start being able to do this for single gene diseases and look at a space of 50, 80, 100 different Mendelian traits, disease traits or non-disease traits, and make predictions about the fetus based on that in a non-invasive, inexpensive, non-risky way. I believe that when that happens, the percentage of women's pregnancy, the percentage of pregnancies in the United States that get tested will go from under 2% to over 50% and maybe over 80%. Issues will follow. The big controversies will revolve around abortion, will revolve around eugenics, and will revolve around disability rights and disabled communities. There will be hard questions about what kinds of tests one should allow for serious diseases, for non-serious diseases, for sex, for non-disease, non-sex traits, for selection for fetuses, ultimately babies who will have some disabilities, maybe the oddest one, but one that's been discussed occasionally in the context of certain disability communities, like the deaf. Legislatures will try to decide when this should be allowed. That could be a good issue for this commission to look into. There are also some less cosmic but important questions. Right now, informed consent for amniocentesis is pretty real in the sense that typically a woman has some time to think about it. Usually this is a result of a screening test for Downs or a family history. If you're going in for amnio and the big needle, you know you're going in for something serious. It is unclear whether women signing a form saying they're getting genetic testing based on a blood draw will know what they're getting and will appreciate the answers that come back on 50, 60, 80, 100 different Mendelian disorders. There will be interesting issues of access and insurance payment. One of the big issues will involve Medicaid. Medicaid pays for 40% of the births in this country, an astounding figure. Medicaid is a state and federal joint program. Some states will undoubtedly pay for this if it becomes clinically acceptable. Other states may not. There will be access differences from state to state and based on income level. Those are ethical questions. And then, of course, there'll be the regulatory and ethical questions of the safety and efficacy of the tests themselves. Second issue, whole genome sequencing for clinical purposes. In May of 2009, my Stanford colleague Steve Quake sequenced himself, actually had his postdocs and grad students sequence himself for $48,000, which counted their time at zero. But he sequenced himself about a year later. Some of us thought, gee, it'd be really interesting to see what we could learn medically about Steve. We published that in The Lancet. There were some really interesting things. There was a bunch of stuff that wasn't very powerful and wasn't very interesting, but he learned he is at probably some heightened risk for sudden cardiac death. When we went through this, we realized there were about 90 to 100 things we thought he should know about. At 3 minutes per thing, that's 5 hours of genetic counseling. I can talk for 5 hours, although you won't let me today. But most people can't. Who's going to do that counseling? Who's going to listen for 5 hours? Who's going to pay for 5 hours of counseling? Whole genome sequencing will raise big questions with accuracy, both of the sequencing machine's test methods, with questions of accuracy of the analysis, of the interpretation. Who decides which variations mean what level of risk? That's going to be an enormous problem and very difficult for us to figure out a useful medical and social way to deal with. There'll be huge questions of the physician's role. Will this have to go through physicians or other healthcare professionals? Personally, I think it should, although we've already heard something about the conflict between paternalism and libertarianism with respect to the genome. Will those physicians be liable for, potentially, I'm a lawyer, liable for, will they have a professional duty to talk about everything that's in the genome or only the thing they're looking for? I worry that at some point this will get cheap enough that if you're interested in, say, whether someone is susceptible to Lynch syndrome, a high colorectal cancer syndrome, it'll just be cheaper to get the whole genome. Well, if you get the whole genome, do you tell them about just the Lynch syndrome? Do you tell them about the 7 autosomal recessive diseases they're carriers for? Do you tell them about long QT syndrome or their ApoE status? What do you tell them about? And how often do you tell them? Their genome won't change, but our interpretation of their genome will change every week. Is there an obligation to rerun the material on some regular basis? How do we inform and educate physicians so they can talk to their patients? Physicians who, for the most part, got almost no genetics in medical school and don't actually have a lot of spare time to learn new things. How, and I think this is going to be the hardest part, how in the world do we educate the patients? How do we convey to them what all this means in a way that doesn't lead them to make a mistake in either potentially dangerous direction? One mistake I am terrified about is a woman is told she doesn't have a high risk of breast cancer, so she decides she doesn't need mammograms. That could be a fatal error. Her risk may not be 80%, but it's gone from the population-wide 12% to about 11.98%. Alternatively, someone is told their risk of Alzheimer's is 2 times normal, and not told that's about 20 to 30%. And most of the time, most people with one ApoE4 allele are not going to get Alzheimer's disease, and overreacts, decides not to go to medical school or law school, instead is a beach bum, pulls out all of its retirement savings, commits suicide, does something else foolish. How are we going to make sure, how are we going to help people deal with this enormous flood of information in a way that's comprehensible to them? There are other interesting questions. What do we do about kids who, as you've heard, the general view is we don't tell them or their parents things that aren't actionable during their minority? What happens if we've got the whole genome sequence? Who learns? Who knows? Point 3, data. What I've talked about so far, the prenatal and non-invasive prenatal genetic testing, and the whole genome sequencing, whole genome or other broad genome sequencing, I'm not sure it'll ultimately be whole genome. It's really only as important as the things we can tell people about. If we can tell people about a lot of important disease risks or a lot of interesting traits to parents, they'll use it a lot. If we can't tell them about very much, they won't use it a lot. But the way we find out what it means is by looking at vast numbers of people and vast numbers of gene sequences and genotypes and vast amounts of clinical data. Well, how do we do that? First, there are questions of privacy. Who all can have access to this? In a way, this overlaps with the forensic discussion, because you don't need a 300 million person CODIS database if everybody's sequence, including their CODIS markers, are in their health records. And the FBI or the local police or a civil litigant, arguably, can subpoena those from the health center. Things like that have happened already. Second, people talk about anonymity. Well, you're not going to have anonymous records for clinical purposes. That defeats the purpose. Maybe you say, well, we'll strip the identifiers out for purposes of research. But that is false hope. Any rich database, any database with health records can be re-identified, almost anybody. Lots of people in that database, you'll be able to say, aha, based on that information, that's got to be James W. Wagner. Only person it could be. Even if there's no name, social security number, et cetera, in there. We are pretending this is not a huge problem. Re-identification is a huge problem. Third big problem here, I think, is the issue of incidental findings. It's been talked about. I'll just say it applies here as well. It applies both in the direct clinical sense, it applies in other research senses. I think the fourth, though, the last, is maybe the most important. Might be something that the Commission can do something about, although it might be too big an issue for it. What do we do about people's consent? If we're really interested in getting all this sequence data and health data, electronic medical record data, useful, incredibly useful potential resource, do we have to ask everybody about each study we do? Do we have to ask them whether they want to be researched on at all? Do we say this is part of your duty as a good citizen, as a beneficiary of the healthcare system, to participate in this controlled work? What we are doing now, I think, is in some ways the worst of all worlds. We are using data broadly. When people don't understand that it is being used broadly, they give it for what they think is a narrow purpose, with a little bit of language tucked in at page 17 of the consent form saying, and may be shared for other research or other medical purposes. And so people who participate in a Parkinson's disease and genetics study in Rhode Island find out, or they haven't found out yet, that their genotypes and phenotypes are in dbGaP, an NIH database which is accessible to researchers all over the world for research on anything they want to be researched on. That question of control is a tricky one. What should we require, and should the culture change in terms of what we require? And I think that's a potential landmine right now, because I think there are a lot of people whose DNA and materials and data are being used for research who don't know what it's being used for, and will be annoyed and unhappy when they find out. So, 3 quick issues. There's a lot more I could talk about, but with 4 seconds left, I'll end.
Thank you very, very much. have opened up here not only what the science does, but what the potential implications are for our role as a commission on as giving advice on the ethics and social responsibility of new technology. So let me just ask you, let me start with Martha and ask Martha a question, because this is the new science. You said one of the red herrings out there is mind reading, and neuroscience is not capable of mind reading, nor is it in the foreseeable future capable of it. So could you say a little, because there is a very big gap in neuroscience, and it's often misunderstood, that the brain isn't the same as the mind. And so let me just give you an example. There's a big issue about causality and how much we can predict and therefore control behavior. So I was the keeper of the clock here, and let's just say I had told you to stop after 10 minutes. I said, Martha, stop. And you said, no, you gave me 15 minutes. And I said, okay, go on. So the reasons you gave me, which is 15, not 10, actually caused me to change my behavior. But the cause were reasons, right? The cause was nothing out there. It was a reason that caused me to change my mind. How does neuroscience understand how reasons are causes? They're causes. I'm not saying that the reasons came from some spooky place, but reasons cause us all the time to change our behavior. And isn't that a limit on what neuroscience right now can do predictively?
Oh, boy. They didn't tell me there was going to be metaphysics at this thing.
It's not metaphysics.
Well, I mean,
there's nothing metaphysical about what I just said.
No, but I feel that there is a sense in which I think a good answer to that question does somewhat get us into metaphysics. But let me sort of
you're treading on my ground.
Yeah. Here's the thing. That hypothetical exchange about the time definitely has an explanation in terms of reasons. It also has an explanation in terms of patterns of neural firing. Okay?
Sure.
And the metaphysics is basically the incredible mystery of why that physical story comports so well with the story in terms of persons and reasons and goals and desires and all those kinds of things. For some purposes, the useful analysis, and you might even say right analysis, is the analysis in terms of people's reasons for doing things. But for many socially important problems, a very useful kind of level of description is in terms of chains of physical events. And in some sense, it's an empirical question whether you can do better predicting behavior with a kind of a reasons-based account or with a neurons-based account.
In this case, there's no doubt that you could do better with the reasons than the neurons. Nobody has a clue as to what neurons caused you to tell me accurately that it was 15 rather.
By the way,
than
somebody
10.
told me 17, but
Right.
I won't out
What?
the staffer. But anyway.
No.
So here's the thing. Yes, so this would be a case where if the question was, is neuroscience going to help us with this kind of situation, managing it better, improving communication, whatever, the answer would clearly be no. But I want to.
That's all I wanted.
But I want to dig in my heels and say that there are many important situations, from marketing to education to security screening and so forth, where those neurons tell a lot.
I agree.
Okay.
So it's not but what I think we've just established is it would be a mistake and futile to think of it as an either-or. That is, if
Absolutely.
you don't integrate the causality of reasons along with the causality of external environments on the brain and the mind, you don't have the full story. That's all.
Agreed.
Okay.
Agreed, yeah.
Yes, Nita.
Hank, I wanted to focus on your third category, the identification category.
Yeah.
And I wanted to look at it from the perspective of both neuroimaging and genetic information. So we have the Human Genome Project, there's also the Human Brainome Project, and attempts to try to do as much neuroimaging as possible to try to see the commonalities across different individuals and also differences, and do a lot of the same types of population statistics that we've done on the genome side with the brainome side. Obviously, there's different information that the two fields can yield, but I wonder if you could speak to some of the ethical issues that you raised to see if you think that there are meaningful differences for data collection from neuroimaging than genetic collection.
I think there are differences. I'm not sure I think they are meaningful. I think everything I said with respect to, almost everything I said with respect to the questions of genetic data could be applied to neuroscientifically acquired data, also applies to old-fashioned health data. That these questions of incidental findings, of consent, of privacy, and of re-identification, the lack of anonymity apply. This could be a good cross-cutting topic for the committee, for the commission. I also think, though, getting back to the issue of forensic writ broad as encompassing not just courtroom use, and not even necessarily criminal or legal system use, but non-medical uses of both genetic technologies and neuroscience technologies. I think there are similarities between some of the marketing issues, whether it's done based on someone's genome or based on somebody's MRI scan, on some of the, certainly on many of the criminal issues, the criminal defense issues, which you've been following very closely. You know, defendants are saying, my brain made me do it. They're also saying, my DNA made me do it. So I think that might be another overlapping area. Whether, as a strategic matter, you're better off looking for an overlap area that deals with both of these or focusing on just one is, I think, a hard question. There are pluses and minuses to both of them. And I'm glad I don't have to make the decision.
Raju.
Thank you for your presentations. They're really terrific. So I want to address a question to Hank. This morning, several speakers really talked about this issue about what should we tell the individual when a whole genome sequence was done, and whether you should inform them only about those findings that are actionable versus those findings that may not be immediately actionable. Can this problem be solved by just asking initially from the individual as to what is it that they would like to have, and would that solve the problem, or there are other types of issues that are very difficult to do?
I think that could help the problem. I'd be reluctant to say that it could solve the problem. For one thing, I think it's a different problem in different contexts. It is a somewhat different problem if you're a researcher who stumbles on something in your research as opposed to the treating clinician. This is your patient who you've ordered this genome test for clinical purposes. I think that has somewhat different obligations and liabilities. I do think exploring in advance before either the research testing is done or the clinical testing is done, what kinds of information the patient or subject would like can be a very useful step. I worry a little bit, though, that the subject or the patient doesn't. It is one thing to answer in the abstract. Yes, I think I'd want everything. No, I don't think I'd want everything. And it may be a very different thing if the patient is then confronted with a very specific kind of disease or a very specific kind of question. So getting some information in advance, I think, can be helpful. It may not always be determinative because the patient's abstract answer might be quite different from the patient's actual wishes or the research subject's actual wishes in a concrete case. Let me give you an example. Subject may say, I don't really care about getting predictive stuff. It doesn't matter to me. But it turns out that if you can predict, say, Alzheimer's disease, this particular patient or subject's parent died of Alzheimer's disease, and Alzheimer's is something that, in particular, this person really, really cares about and would, even if there's no intervention, like to know about. When she answered the question up front in the abstract, she wasn't thinking about Alzheimer's disease in particular. Maybe if you've got Alzheimer's findings, there should be some re-exploration of the specific kinds of findings you might be able to provide. I do think it's the sort of thing, and this is, of course, one of the huge reasons why I think direct-to-consumer information is so risky. If it's only SNP chips with the kinds of fairly weak associations that they've had right now, there's not all that much of power. But if you get to whole genome sequence, and there are very powerful associations, I think almost everybody is going to need a qualified, skilled professional to help them understand what this means.
Thank you. Anybody in the audience have a question before we take a break? I see a hand back there, yes. And please introduce yourself. Just tell us who you are. Thank you.
And I'm just here because I'm interested. My question is for Dr. Farah. You mentioned that we're a long way from mind reading, yet a couple of years ago I saw on the History Channel they showed a mute paraplegic where they had implanted a chip in his throat, and his thoughts were going onto a computer screen. Maybe I'm confused as to the difference between mind reading and being able to capture human thoughts onto the screen. I'll let you respond.
Great question. Thanks.
Okay, well, I'm thinking there are two possible technologies that you can, again, it's very strange to talk to somebody with your back to them, but forgive me, Lisa. There are two possible technologies that you could be talking about. One does sense microscopic movements in the vocal apparatus and basically looks at subvocalization. So it's basically a way of just using speech that you can't hear. I think that's a cheat. It would be a cheat to call that mind reading. There have also been some early human trials of chips implanted in the brain on parts of motor cortex that allow a paralyzed person to move a robotic arm or move a cursor on a screen or type a message on a computer screen using thought alone, by basically learning to kind of think about movements and direct the cursor or the robotic arm accordingly. There is a sense in which that's mind reading, but consider this. It's basically translating brain activity into movement in, at most, three-dimensional space. Okay, non-trivial, but to read a thought like Hank is thinking now, oh, poor Martha, she blew it again. She went over, and the chairman had to tell her to end early. That kind of thought, if you think of it as a spatial analogy, is a point or a path in an extremely high dimensional space, okay? All the different dimensions of meaning and intention that a person could conceive of. That is what we are a million light years away from doing.
Hank's actually just thinking it's too bad for Martha that the chair of the commission is the president of Martha's University.
But I'm
Happy
thinking,
for me,
Martha,
not mine.
Not only do I admire Martha, but she has tenure as well. Thank you all. We're going to reconvene. This opens a host of issues, and it gives us a lot to think about, whether our minds can be read or not. And also, you don't need to read our mind to know that we will reconvene at 1:15 promptly. So thank you all very much, and thank you again for wonderful presentations.